Just wanted to say thanks for providing the opportunity to present. In the next 30 minutes, what I'm going to talk about is the confluence of innovation in therapeutics, drug delivery, and regulation. So the talk is going to be divided into two sections where I will briefly go over the novel modalities, drug delivery technology. We'll take a look at some regulatory guidance that's currently available and how can we leverage this for the newer modalities that are in the market. And what should sponsors do when you have modalities that are not antibody-based? So how do you approach that? So that takes us into the third point where we will look at regulatory considerations that may address future issues with novel modalities and drug delivery technology. So this will be more like an industry perspective. So if you're in the shoes of a company where you are probably the only expert in the world, how do you approach this just to make sure that innovation and regulation are not out of sync? So let's take a look at, the innovation in therapeutics. So as you can see here, the innovation in therapeutics has moved at a lightning speed. So the industry started off with small molecules. So collectively, we have a lot of experience in small molecule or synthetic development. These are chemical molecules. You have a very set process. So when you compare this with the other modalities currently on the market, it seems much easier to handle this. That being said, there are very specific issues and it can also be very challenging to manufacture this. So definitely wanted to point that out as well. We go to the second picture here in the middle that says therapeutic proteins. So these proteins are large, complex proteins that are manufactured in living cells. And there are multiple points of failure. You can have all the processes and controls, but at the same time, there's always that possibility that things may fail. From a regulatory perspective, these proteins need to be characterized quite a bit. So there's a significant regulatory data package that needs to go in. And if you look at the last bucket over here, we have novel treatment modalities. So these modalities, some of these are antibody-based, so they may be using that framework and now they're built upon it, or you can have cell and gene therapy. So the novel modality bucket is really wide and it's very varied across the board. You can use experiences from the mAb-based regulatory or institutional knowledge, but at some points, you will come across where none of this knowledge is applicable. And now you're at a point where you really have to work with the regulatory agencies to make sure you have the regulatory guidance that you need. So again, at the end of the day, trying to work with the regulatory agencies to make sure you're not out of sync while the innovation is going at lightning speed. All right. So let's take a look at the innovation antibody-based therapeutics. Due to the time considerations, I definitely won't be able to do justice. So this is, please keep in mind, this is just a subset that we're going over. And this presentation is based on a publication that came out last year from our group. So if we take a look at the bispecific and multispecifics over here, the concept of this has been around since the 1960s. So if you take a look at the diagram over here, which is from one of the Nature articles, the concept existed in 1960s, but by the time we had something on the market, that's, if you look at the arrows and follow through, it's 2017. So in between this time, there has been a lot of innovation that has gone out, that has been going around. And that relates to making sure how can we come up with these stable bispecific or multispecific formats. So conceptually, something may look easier, but to put it into practice, to have a clinical molecule or commercial molecule, that takes significant effort. And there's a lot of innovation, collaboration with the regulatory agencies. So if you look at it, there have been 100 bispecific formats that have been reported. More than 20 of these have entered the clinic. And commercially, at the moment, we have 2, Blincyto and Hemlibra. Okay, oops, sorry. So in terms of challenges, there's a lot of potential for these multi-specific and bispecific modalities, but there are a lot of challenges as well. And most of these challenges come around the manufacturing, but they're not just limited to it, but most of them are around the manufacturing. So if you look at it, you're taking two different antibodies, you're trying to combine them to make one molecule with two separate binding targets. Like I said, conceptually it looks easy, but it's not as easy. Then you also have to figure out if you have issues around the clinic. So will you have a clinical cytokine release syndrome? How will you deal with it? What would the dosage form be? And how will you set up your clinical trials? So bispecifics, if you really look at it, so these are molecules that are targeting two different targets, and that's very different from your regular monoclonal antibodies. So the next modality we can take a look at is this masked antibodies, and these are supposed to go after the undruggable targets. So the concept around here is you have a antibody-based format, but if you look at the diagram on the right, you have these little bars, and these are supposed to be the masking peptides. So the idea here is the molecule will not be activated until it reaches the tumor-specific environment, and it gets activated by the tumor proteases. So really neat concept, and definitely helps with those undruggable targets. And what that means is if you have a target that's expressed in multiple parts of your body, if you have a molecule against it, that means you also have a lot of off-target toxicity. So this modality is supposed to help with reducing that off-target toxicity. So lots of potential, but there are challenges as well. And the challenges come around this masking peptide. You have to make sure you have a target-specific mask. You also have to make sure that it's tight enough and it doesn't come off loose at any given point during administration or during any other environmental stress. Because if it does, then that means you will have a lot more off-target toxicity. Sort of defeats the purpose of having this modality. And there are a couple molecules in clinical trials. So time will tell for the true potential of this modality. Okay, next slide. So this one is the single domain antibody fragments. So until now, we talked about modalities that were monoclonal antibody based. These are 150 kilodaltons. So these are huge molecules. That can be used for metabolic indications and for cancer therapy. But when you really look at using monoclonal antibodies or proteins that are as large as these antibodies, and when you're targeting them for a solid tumor, they have very poor vascular permeability. So they may not be the best. So in that case, single domain antibody fragments are perfect because these are 10 times smaller than your regular antibody. They have great vascular permeability. That means this modality can be perfect for solid tumors. FDA approved its first single domain antibody fragment in 2019. So the size is an asset, but it can also work against us because the smaller the size, the faster the molecule can be cleared from the kidney. So what that means is you may need additional doses or you may need longer treatment for patients in the clinic. So again, there's a lot of potential, but at the end of the day, you really need to make sure as a sponsor, how can you deliver a therapy that is effective, but also convenient to the patient. And again, for all of these that have mentioned, regulatory guidance from the antibodies definitely can be leveraged. There will be specifics which will have to be worked out, but that's where the regulatory agencies can definitely come in and provide guidance if additional guidance is needed. So let's take a look at another scaffold, which is a protein, which is a protein therapeutic scaffold. So this is a non-antibody based scaffold. And these are very thermostable. Again, they're 1/10 the size of a full-length antibody. And these are called DARPins, design, anchor, and repeat proteins. So in terms of yield, they're also easier to manufacture. You can get about 200 mg from 1 liter of culture. There are a couple in the clinical trials, so time will tell how effective this scaffold therapeutics are. But if you look at the diagram on the right, you have an ability to make multiple and very bispecific or multi-specific formats with this protein scaffold. You, of course, will need linkers to make sure to connect these different targets. And that's where the challenge could be, because anytime you have linkers, you want to make sure they're stable enough. You don't have proteases which are clipping the linkers, because then it really defeats the purpose of having these. So again, there are challenges, but there's potential as well. And like I said, clinical trials will let us know what is the true fate of this modality. Okay, let's take a look at multifunctional modality, and these are PROTACs. So these are nature's way of dealing with unwanted or mutated proteins. So humans have a natural way of disposing proteins that are not needed through the proteasome. So as you can see on the slide in the diagram that's right here in the middle, you have E3 ligase that recognizes the protein that's no longer needed, and it repeatedly tags it with ubiquitin. So that basically signals that this protein needs to go to the proteasome and it needs to be denatured. So PROTAC is a small molecule and has two binding regions. So on one side, you have a protein that's targeted for destruction. On the other side, you have the E3 ubiquitin ligase. And both of these ends are joined by a linker. So here's an example of a linker. So these have multiple routes of administration, and they have the potential to target the undruggable. Basically, proteins are targets that are expressed multiple times in your body, but if you have a drug against it, you could have a lot more toxicity effects. So these PROTACs are perfect for that sort of the undruggable. There are a couple in clinical trials, and so hopefully we'll know more as we follow the clinical trials in terms of the future of this modality. But just like the DARPins, you also have a lot of potential here in terms of developing bispecific or multi-specific formats. The challenge with PROTACs is this is really brand new. So we have a lack of historical precedent in generation, optimization, clinical and commercial product development. You will need definitely extensive product characterization from a regulatory perspective, and it could be expensive to manufacture as well. And these are going to be more complex than your monospecific drugs. So at the end of the day, again, balancing out the pluses and the minuses, and then working with the regulatory agencies to see what additional information is going to be needed for a product to be on the market. Okay, I definitely couldn't present without mentioning cell therapy. So like I said, I won't be able to do justice, but I definitely wanted to mention. So this is one of the categories or the modalities you can consider where the innovation and regulation can go out of sync, because the innovation is happening at such a high, at lightning speed, at a really high pace. So if you look at the cell therapies, these can be divided into two major buckets. One is the autologous cell therapy, where the stem cells are harvested from the patient and they go back to the patient themselves. But there's another part where is the allogeneic cell therapy, where you can take patients, or where you can, take stem cells from one patient, but then you can, the culture can be expanded, and now it can be used to treat many patients. So the problem here is, and the challenges are, again, focused on manufacturing, release testing, cost of therapy, and at every point, if you're a sponsor, you definitely want to make sure you are in touch with your regulatory agencies, figuring out which regions of the world are you planning to launch your product, because things will change accordingly. And from manufacturing perspective, you really want to make sure that if, especially for the allogeneic therapy, if you have an individual and you have some sort of latent infection, you want to make sure that gets caught early and you don't amplify that. And now you're introducing that to a lot more patients. So as effective as this therapy is, it also comes with its own set of challenges. And if you look at the regulatory guidance, so this is a perfect example where your regular monoclonal antibody guidance doesn't really work. The cell and gene therapies are its own bucket where it needs very specific guidance from like regulatory agencies. Okay, so switching gears, hopefully at this point I've given you a little bit of an idea about the different modalities that are out there in the market at the moment, and the potential they have, and a little bit idea about the challenges as well. So that's focusing on the modalities, but on the other side of drug development is always the drug delivery aspect, and how can you make sure that the drug is delivered in a sense where it is the most convenient for the patient. So let's take a look at a couple of drug delivery technologies. So first up is co-formulation. So this has been around for a while, but recently you have seen some products on the market. So what is co-formulation? Anytime you have drugs that are put in the same dosage form, then that defines as a co-formulated product. So this can be two new entities, or it can be one new entity, one commercial product, or it can be two commercial products. Either way, it'll define as a co-formulation as long as they are in a single vial or a single dosage form, as you can see in the diagram in the middle over here. The only challenge is you have to demonstrate superiority to monotherapy. So you can't combine products just because it's convenient. You have to establish that combining these two products is superior than injecting two of these products individually. So shown on the bottom right in blue text, I have a couple examples, and a couple of these actually have the potential to increase bioavailability with soluble enhancers, and that's the Rituxan Hycilla and the Herceptin Hylecta. So this is an example of a patient-centric dose where if you have non-obligatory targets, you can combine them into a single dosage form, and you can, so the patient doesn't have to come back. It's convenient for the patient, so it's more patient-centric. But on the challenges side, again, you will need an extensive analytical data package and make sure the drug product is analyzed and completely understood, because anytime you mix two products into a single dosage form, you could see a lot of protein-protein interactions, viscosity challenges, method development issues. So all of that has to be worked out. And again, constant collaboration and dialogue with the regulatory agencies will really help if you have a co-formulated product or you're planning for one of them. Okay, oops, sorry. Another one is lysozymes and nanoparticles. So this is a very diverse area of drug delivery because it can deliver a unique range of drugs. Pretty much everything can be delivered through lysozymes and nanoparticles. The current COVID-19 vaccine is actually part of this, the lipid nanoparticle vaccine. So there are 4 types of delivery systems. The first one, figure shown here on the right, type A is your traditional lipozyme. B is the pegylated, where the peg is added to increase stability and also the circulation time. And the C is a very interesting one because now you're really getting into very targeted therapies. This is the ligand-targeted lipozyme. And the fourth is actually a combination of all 3 components over here. So like I said, you can deliver small molecules, you can have biologics as well. But again, the challenges are still around coming up with a homogeneous product. There's a lot of cost and coming up with a very specialized clinical strategy. And last but not the least, liposomes have been around for more than 50 years. So any sponsor will definitely run into the intellectual property issues. So as long as all of these can be figured out, and if you have the right target and the patient population, then this seems like a pretty attractive way of delivering drugs. Okay, oops, sorry. I keep moving to the next slide. The next one is inhalation and pulmonary delivery. So I've combined both of them in here. So if you look at this delivery, this is more like a organ-specific delivery. This can be applicable to small molecules, peptides, and proteins. So inhalation delivery is a non-invasive alternate drug delivery route, especially for patients who may be afraid of needles, for example, and if the indication is, say, in the lungs, then this may be a route to go. Also, from a biology perspective, if your target is within the lungs, then you can have rapid onset of action for pulmonary delivery. So usually you have devices that are combined with it. You will use like an inhaler or a nebulizer to deliver the dose. So if you're using any sort of small molecule, peptide, or protein, you want to make sure that your product is still stable. The stress that it sees going through the inhaler or the nebulizer is not compromising the product. There's another challenge as well, because from an anatomical and immunological barrier, especially in terms of biologics. So it's not really easy to have full-length monoclonal antibodies being delivered through the pulmonary route. But like I mentioned earlier in the presentation, we have a lot of modalities that are coming that may be 1/10 the size of an antibody. So that means it may be easier to deal with these modalities and have them deposited in the lung. It may be easier compared to a full antibody, which is like 150 kilodaltons. So there is potential. And again, this very specifically is going to be dependent on the interplay between the modality, the device, the patient compliance, and the regulatory approval. Okay, last but not the least, oh geez, my mouse is really sensitive. We have the 3D printing for a patient-centric therapy. So currently this is for small molecules or synthetics. So 3D printing is basically having your drug and it's printed layer by layer compared to your traditional tablet manufacturing. So 2015, a 3D tablet was approved by the FDA. This again provides you an example of a agile manufacturing. So if need be, 3D printing and the facility can be moved really closer to the patient. So instead of manufacturing the drug at a single source manufacturing facility, now you can move or have these smaller agile manufacturing facilities very close to the patient. The trick is going to be to make sure that you establish product compatibility, to make sure that the product is exactly the same irrespective of where it's being manufactured. Not only that, basically showing that there is no difference in efficacy or stability when it's manufactured the traditional way versus the 3D printed way. So again, this is going to be a really neat way of delivering drugs. Currently, it's only limited to tablets, transdermal patches, and vaginal delivery systems. But again, there is potential for this technology. Okay. Okay, so I mentioned, so hopefully I've given you an idea about the novel modalities that are around in the biotech industry right now, the drug delivery technologies that have a lot of potential, and potentially these also can be used for these newer modalities that are a lot smaller in size than your regular monoclonal antibodies. So regulatory guidance, and I'm just giving you an example of the monoclonal regulatory guidance that exists here. So it's very specific. There's a lot of characterization that is needed. A lot of this can be applied to the newer modalities that, like I mentioned before. Of course, cell and gene therapies, that's its own bucket, and those regulatory guidances are very different. But if you are a sponsor going into coming up with newer modalities or drug delivery technologies, then it is really important that you're in constant dialogue with the regulatory agency. And how, so this is the industry perspective that I was talking about at the beginning of the presentation. How do you approach this? So there are different ways of doing this. You can interact with them on a one-on-one meeting basis, but you can also leverage the conferences, the consortia efforts, or the workshops and webinars to educate the industry and also educate the regulatory agencies. especially for brand new modalities and the innovation that's going on. It's going to be really tricky because at any given point, your company or that subset of people may be the only folks who really understand this technology. But from a regulatory perspective, eventually the drug will go to the clinical stage, the commercial stage, and it will need approvals. But at the same time, you also want regulatory agencies to understand your concept and your innovation. So for that, the earlier you work with the regulatory agencies, the better off you are to successfully launching your product. Of course, all of this depends on how much you can reveal, how much you can talk about in the public space, given the intellectual property that's always going to be part of these innovation. So the second bullet point is considering the CMC consideration. So looking at what can you leverage depending on your modality that's already existing in terms of regulatory guidance or institutional knowledge that can be leveraged for the CMC. That would really help. And areas where this cannot be leveraged, you will be coming up with newer regulatory data package and requirements to support it. So that's where it will, again, really help when you have that constant dialogue with the regulatory agencies to understand what is needed for your drug to be safe, efficacious, but at the same time approved by the regulatory agency. And last but not the least, the structured content and data management solutions. Biotech has gone global. The teams are all around the world, and that is true with the regulatory framework as well. So in having access to data for scientists as well, but also for the regulatory agencies, if you can do it like a real-time learning mode or have access to real-time data, that can also speed up your regulatory submissions or just that conversation around what is really needed for that regulatory data packet. So that would help as well. So basically, hopefully I have given you an idea that we have a lot of novel modalities and drug delivery technologies in the clinical trials at the moment. Despite excitement, there are challenges that exist, and there are regulatory challenges as well that need to be addressed. We can also look at potentially evolving regulatory paradigms for a single worldwide dossier using digital platforms where we can be feeding in live data. And not only the scientists, but the regulatory agencies also has access to this data. So if there are any questions that arise, it may be much easier to address this, especially to match the pace of the innovation. So I would like to conclude the talk by saying it's really critical for the industry and regulatory agencies to collaborate towards a confluence of innovation and regulation to accelerate access to life-saving medications for patients. So before I end the talk, I wanted to make sure that all the references that were mentioned in the talk are listed over here, and I'll be happy to share this if need be. Like I mentioned before, today's talk was based on the JPharmSci publication in 2020, late 2020. And before I end the talk, I would like to give a shout out to my co-authors, Lucas Nina, Mike Giffen, and Mike Abernathy for a great collaboration. And thanks again for listening to my presentation.